Thank you very much for the introduction. Forgive my voice, I don't always sound like this. But I'm also going to talk about invasive blue catfish populations in the Chesapeake Bay region. But I'm going to talk about a different aspect of blue catfish biology. Specifically, I'm going to talk about reproductive biology of blue catfish. Just so I know, how many people here did not see Corbin's talk, just the one before this? A few. Okay. All right. So, as you know, blue catfish are freshwater fish native to the Mississippi, Missouri, and the Ohio River basins. But they were introduced in the Chesapeake Bay region in the 1970s and 80s to create a recreational fishery. They were basically stocked in these locations highlighted by the green triangles. But since then, they have expanded quite a bit in range throughout the Chesapeake Bay region. So all of these brown areas are where blue catfish occur now. In addition to this range expansion, there has also been a tremendous increase in the abundance of blue catfish. This is showing the index of abundance over time for James River and York River in the purple dashed line, based on the Vim's Juvenile Fish Trawl Survey. And this essentially shows that the index of abundance has increased tremendously in both James and York Rivers. For the York River, it may not be as clear, but that's just because of the scale. If we zoomed in, then we would see that that has increased in the York River as well. So concurrent to this increase in the abundance of blue catfish, there has been a decrease in the abundance of white catfish, which is the native catfish in that region. So the abundance of white catfish decreased precipitously in both of these systems. And this is just to show that blue catfish may have negative impacts on some of the native species, such as the white catfish, because of competition, but this is just an example. So blue catfish might have negative impacts on other estuarine animals, such as the blue crabs and the alocins and so on. So blue catfish are now considered an invasive species in Chesapeake Bay region, and we care about invasive species because they harm our lands, waters, and native species. So there is a huge interest in managing blue catfish in the Chesapeake Bay region, and this is sort of the framework that often people use for any kind of fisheries management. So you have population biomass. There are two fluxes that increase population biomass. Those would be growth and immigration. Two fluxes that decrease population biomass, mortality and immigration. So assuming that immigration and immigration are sort of constant, then we focus mostly on growth and mortality. Growth, again, can be divided into growth in terms of size and growth in terms of recruitment number. Mortality can be divided into natural and fishing mortality. So management focuses on fishing mortality. But to know how much fishing mortality to subject the population to, essentially we want to know, how much fishing we should do to take the population biomass where we want it to be. But to know about that, you need to know about all of these other factors. We have some rules of thumbs to know about natural mortality. Size, increase in size, Corbin just talked about that. Then the other aspect is recruitment, and that is one of the trickier ones that we do not really have much idea about. So recruitment essentially comes from two factors. One, the reproductive output itself, the number of offspring produced. The second is the survival of the offspring. I'm going to talk about the former, the reproductive potential of blue catfish. Then I also wanted to assess the population-specific differences in reproductive traits. Why did we want to do that? Because these populations have very different population dynamic rates. For example, for the James and York Rivers, you note that the James River has substantially higher index of abundance compared to the York River. And that has important implications for life history traits. All right, so here I am showing age in the x-axis, length in the y-axis based on life history theory. You already know that these curves are probably going to look sort of like von Bertalanffy growth curves, right? So the blue line shows a population at low density. The dashed orange line shows population at high density. The circle and the triangle correspond to maturity events. So compared to high density, the fish at low density grow faster and they mature at a larger size but younger age. This is what is predicted based on life history theory. and also pretty well supported by a lot of empirical studies on a lot of species. But in any case, at this low density, they mature at an earlier age, but after that, they allocate most of their energy into reproduction, so they do not really grow much after that. In contrast, at high density, they allocate most of their energy still into growth so that they can continue growing large and eventually reach the size as the fish that were growing at low densities. And they want to do that, I say want, they do that because growing larger is better for two reasons. First, if you are larger, then you are able to produce more eggs. That's always good. The second aspect is if you are larger, then you are less likely to be preyed upon. So those are the two reasons this usually happens. Is this what we observed in the James and York River fish? That is what we wanted to assess. So we went out to the tidal James and York Rivers, collected blue catfish using electrofishing for the most part, but also some other gears opportunistically. The fish were brought to the lab, and we collected data on length, weight of the fish, weight of the ovary, and so on. We also got the ovaries, we also got the otoliths, which we used to age the fish. For the reproductive traits, we had four main reproductive traits that we looked at. First, we looked at gonadosomatic index, which is just the proportion of body weight that is contributed by the ovary. Second, we assessed the maturity status, whether a fish is mature or not. The gold standard way of doing that would be histology, and we tried that, but we were not successful consistently. So we had to rely on another approach, which is based on gonadosomatic index. This idea comes from this paper by Fontoura et al., who essentially developed this rule of thumb that said that if the gonadosomatic index of a fish is 5% or more of the maximum gonadosomatic index, then you can consider that fish mature. Of course, this only applies for fish right before the spawning season, because after the spawning season starts, some of the fish may already lose some of the ovaries, some of the eggs. So this approach actually has been found to work pretty well in multiple species. Multiple papers have shown that, so we feel pretty comfortable with this. We also assessed fecundity, which is just the number of eggs produced by each fish. To do this, we use the standard gravimetric method. Essentially, you take a small subsample from the ovary, count the number of eggs in that subsample, and then extrapolate that to the whole ovary, well, both ovaries. We also assessed a couple of egg characteristics, and I'll talk about that very briefly at the end. Jumping into the results, here I am showing gonadosomatic index in the y-axis, month in the x-axis, the top panel for the littlest fish, less than 400 millimeters, the bottom panel for the largest fish, greater than 600 millimeters in fork length. Two things to note here. First, you note that the gonadosomatic index is highest during, say, April, May, June, July. This essentially says that probably that is around when spawning occurs. The second thing you note is that as you move from the top panel to the bottom panel, you note that in the top panel, during April and May, the gonadosomatic index is relatively low in most individuals. June and July, the gonadosomatic index is much higher. Compare that with fish, the larger fish, where the gonadosomatic index has declined tremendously by July. This essentially means that larger fish spawn earlier during the spawning season. And that has important implications, I think, because depending on what your management goals are, when do you harvest fish of what size during the year? That has pretty important implications. Now let's look at maturation schedules. Here I am showing probability of maturity in the y-axis, fork length in the x-axis, blue for the fish from the James River, orange for the fish from the York River. First of all, you note that as fork length increases, the probability of maturity increases as well. That's a given. Interestingly, the rate at which the probability of maturity increases for the James River versus the York River are actually different. Fish in the James River mature at a smaller size compared to the fish in the York River, and that's statistically significant. This is based on logistic regression. So, fish in the James River grow slower, they are at higher density, they grow slower, they mature at a smaller size. When we look at A's, the pattern flips. fish in the James River mature at an older age compared to the fish in the York River. And both of these things are as predicted by life history theory, as I showed before. Also, I should mention that these ages, 7 and 7.7 years for age at maturity, mean age at maturity, seems to be higher compared to what has been observed in the native populations, and that might be because of higher densities of this species in these rivers compared to the native populations. Now let's look at the fecundity trends. I'm showing fecundity in the y-axis, fork length in the x-axis. You note that as fork length increases, fecundity increases as well, and this is going to become important. And I mentioned this earlier as well. The other thing you note is that fish in the James River have higher fecundity compared to the fish in the York River at any given size. Fish in the James River have higher fecundity. This is going to come important later on. But if we can combine this fecundity trend with the length at age relationship, which I'm not showing here, but Corbin just talked about this a little bit. If we can combine this, then we can get an estimate of cumulative fecundity at different ages. Now note here that the fish in the York River have a higher cumulative fecundity at any given age compared to the fish in the James River. Fish in the James River take all the way to age 25 or so to catch up to the cumulative fecundity of the fish from the York River. That essentially seems to say that fish in the James River may not have the same kind of reproductive output and same kind of individual fitness compared to the fish in the York. But that may not be the case because most fish do not live all the way to 25, right? So that may not be the case because that is when the egg characteristics come into effect. We found that the egg diameter of blue catfish were pretty similar between the two populations, about 3.1 millimeters. But when we looked at the egg organic matter, essentially the ash-free dry weight of an individual egg, we found that the organic matter content was higher in the fish in the James River, essentially meaning that the eggs from the fish in the James River were higher quality. Maybe they have greater hatchability. The offspring, the larvae that are produced, are more likely to survive because they are better provisioned. So that is what we have found here. But going back to this fecundity trend, here I'm showing that the fish in the James River have higher fecundity at any given size, and they have higher organic content. This essentially shows that fish in the James River have higher reproductive allocation, but that is not what we predicted from the life history theory. We predicted that at higher densities, they would allocate less energy into reproduction and they can continue growing. But what we observed is that they allocate more energy, which means that they would be smaller eventually, right? Is this what we observe, and why would that work? Two reasons. First of all, as I said before, the two benefits of being large and continuing to grow are that if you are larger, first of all, you are less likely to be preyed upon. In these systems, they are top predators. Nothing really can eat them after they are a certain size. So that is not something that is going to prevent them. That's not a limit. The other factor is, if you are larger, then you produce more eggs. But they are already producing more eggs at any given size. So essentially, they are using this novel tactic that may not be fully supported by life history theory and has not really been observed in a lot of species. They just use novel tactics compared to most other fish species. And that is, I think, one of the take-home messages of this presentation. So to conclude, bigger fish spawn earlier in the spawning season, and that has important implications for fishing regulations. Reproductive tactics and traits differ between populations. We found that the tactics used by blue catfish in these systems are actually novel compared to predictions by life history theory, at least some parts of it. We found differences in maturation rates, fecundity trends, and egg quality between the two systems that we studied. And that tells us that because of these differences in life history traits, harvest strategies need to be population specific to account for those differences. And if these differences occur in these two systems that we studied, it is likely that such differences exist among other populations in the Chesapeake Bay as well. So we need to look at that before we can formulate a management plan. management plan for Chesapeake Bay blue catfish cannot really be, one plan for the entire bay. You need to consider differences among the populations, one way or the other. I think that is the main take-home message of this presentation. With that, I want to thank a lot of folks that helped me process the samples: Jack Buchanan, Melissa Punt, Matt Oliver, and others. George Strice was instrumental in collecting the samples for me. Aaron Bunch from Virginia Department of Game and Inland Fisheries, and VIM's Juvenile Fish Trawl Survey crew also helped me collect the samples. I also want to thank VIM's Office of Academic Studies and Virginia Sea Grant for funding my research. And with that, I'll take any questions that you may have.